computer, share screen, get up, have to come out first, get that. Where did the PowerPoint go? Oh, there it is. So where we were? Okay. How's that? Good? Anybody here that didn't get a PowerPoint? All right. IgM, remember the biggest 900,000 Daltons, but only 5 to 10% of the serum immunoglobulins. Half-life, much shorter, 6 days. What was our half-life for GG again? IgG? 23. Okay, what was that? 23. 23, yeah. But we can remember that the IgM is not always found in the pentamer. We've already seen it in the monomer shape as it came across that B cell as it went into helping the B cell maturation. So here's the pentamer. We see the joining chain, the joint chain linkage for disulfide bonding between two adjacent monomers. Okay, so if you look here, you can see that this is, disulfide bonding is between the separate immunoglobulins, and then the J chain keeps it in that pentamer shape. So the biggest thing here is it has 10 antigen binding sites. So if we're doing antigen antibody testing, again, to remind you, this is the one that's best because it has 10 sites for binding the antigen. So if we're looking for a reaction, we see that easier if we use an IgM antibody against the antigen. So it has a high valence, found mainly intravascular. So this is not one that's going to find its way out of the vascular system. And the biggest thing is it does not cross the placenta, only the IgG. So all the ones we're going to talk about from here in are do not cross the placenta. And then we're going to talk about this primary response. It's known as the primary response antibody because it's the first one to show up. Okay. So when you have an antigen invasion, an immunogen invasion, the IgM population increases in response to that antigen. Okay, so it's kind of like the first soldiers that walk in, okay, and it's synthesized only as long as that antigen remains present. So we have an antigen present itself to our bodies, IgM is developed. The antigen, if it goes away, IgM goes away. So we see this decrease in IgM against the antigen. So take that to today, right? In this current situation we have now, we say, oh, we're getting the antibody test. Is everybody familiar with that term so far with COVID? Oh, we're going to go get it just to see if I've been exposed to it already. Maybe I got exposed in February. Maybe I've already developed an antibody. This is not the one we look for. Okay, we do not test IgM for past infection. IgM is current infection. Okay, so we do that. The doctors will say, I want antibody levels on this patient. I need to know what's have they been exposed? Do they have a current or do they have a past exposure? So we have to, as the immunologist, we have to know IgM, IgG, because Quest or AEL or whoever we're sending this test off to just says, which one do you want? Okay, and they'll have a little, hey, no, this doesn't indicate a current infection. This indicates a current or past infection with IgG. But IgM is the one that would be a current infection. You wouldn't want to run this if you were like, we think they got exposed to COVID in 2020, and now it's 2023, and we want to see if they have immunoglobulin to it before it comes back or another version comes in. So keep that, that's key to the memory bank. And then there's no memory cells. So just IgM comes up, takes care of the infection, goes down, okay? It's gonna rely on who for this, which immunoglobulin is gonna keep this going for us. Just talked about on Friday. IgG is the memory, okay? IgG, we're gonna see it respond as memory cells, okay? So the IgG remembers that infection. So that's why we do the IgG test. So when we test a patient, if a patient's wanting to know, like I did, I wanted to go to Red Cross, I donated a unit of blood, do a free antibody test, right? Yes, we do. So all the blood services are doing antibody testing against COVID. So I had my little, got my little funny app here from Red Cross, like everything has an app, right?
Do they do it automatically or do you have to ask for it?
It does automatically. So it's part of just donating a unit. So I get to go here and it says COVID-19 antibody test, view your results from your last donation. Okay, so I get to post that big positive there. So I got a positive antibody test. So this made my what? My anxiety yes, I got my nasal swab and I tested positive for the RNA, but I wasn't sick. So it was kind of like, is that a true positive? Did they goof something up? What's going on? It could be any of those things. Heard this testing stuff's not very good, right? We've heard all these different stories. But that helped, okay? That helped say, right, you had it because your body mounted a response, your body produced antibody against it, and now we have hopefully memory, right? That if I get exposed again, then I have an even better response to it.
So what's the point of testing for it if they're going to donate it anyway? So like they just do it to do it? Or?
No, what they're wanting to do is they're going to get my plasma off that donation. I gave a whole unit, so they got red cells, plasma, platelets, they got it all. So what they're wanting to do is called convalescent treatment. So if they have a patient that's at NEA or St. Bernard's that's in the ICU and struggling to survive, they can call Red Cross and say, we need some plasma that has COVID antibody. So it's a way of treating a patient passively with somebody else's antibodies to help them get over it. So what my antibodies would do, they would infuse my plasma to the patient. My antibodies would be there and it would start finding the virus and start to stick to it, bind to it, and then their immune response would respond to that immunoglobulin being there. Okay, so it just helps.
So that's what they're doing now? I mean, it's helping?
Yes. Yeah, we one of our many webinars I watched over the spring and summer was California's got ahead of this. They've already proven that 25 of their ICU patients are getting sent home because they have been aggressively relocating recovered. Okay, so you see a lot of, I didn't get on TV, but you see a lot of movie stars that are they're like, oh, I'm giving my plasma now. I know y'all didn't know, but I did have it and I'm over it now. So I'm doing the right thing and I'm donating plasma for it. So what they're wanting to do is get everybody into blood donations best we can and get the test done. And then they could locate maybe 5-6 more people in case they do have an increase in ICU patients. So that's kind of how the story goes. And you need to know this part. So this story beginning for you is just knowing the difference between IgG and IgM. And how can you communicate that to people that ask you, what does this mean? Why is Red Cross and Lifeblood and Vitalant and all these blood services testing for this? Why are they doing it? And it's because they want to locate the plasma units to say, Hospital B, I've got some plasma that's COVID antibody positive. If you need it, we'll send it to you. to treat your patients, and they'll ask for it. Because plasma's frozen. You know, they freeze it, put it in a freezer in the lab, and they'll get it out, and you keep it until they need it out of that patient. So it's not gonna go bad. So it'd be a great thing to do is kind of stockpile, you know, units of COVID positive antibody plasma. Okay, so next semester, I don't know how much you got in principles, but you'll start to learn which units you can donate plasma to different people because of their blood type, right? Y'all done that yet? You done the little cross where you figure out who has antibodies with yours and not your blood type and all that? It's coming. So here's our classic primary versus secondary response. So the first exposure. So this is what's been talked about so much with COVID is we don't know exactly what that time is, right? They push it to 14 days. Well, somebody could be exposed to time 0 and 2 weeks later they develop the illness. Well, firsthand knowledge, we had another wedding. I hate to tell you that, but yeah, I went to a wedding Saturday. Another one. That's where I got it the first time. Went to another wedding Saturday. Scary, right? But we know that last time we had people attend the wedding in June, the end of June, were showing symptoms Monday. Okay. So guess who comes down the stairs this morning to tell me she has a sore throat? Be my daughter, right? So I'm like, well, you just went to a wedding Saturday. We'll watch your fever tonight. Make sure you're not running anything, but it's not 14 days. So don't get in that habit of thinking, when was my exposure? Because I did that. My sister tested positive Monday after the wedding in June. And I said, well, you had to have gotten it before the wedding because it hadn't been time enough. Now we know, right? And that's the info they're not giving back from cases. I don't know why they're not doing more case studies as we're in this so that we can get a better feel for what's going on, right? I mean, we're starting to see things like only 6% of the people dying from COVID are actually only dying from COVID, right? 94% of the people are dying from, they've got other issues, right? They've got heart condition. That's not to say you shouldn't, like, oh, well, they would have died anyway. No, I mean, cancer, heart disease, diabetes, all of that bad health, right, is 94%.
Isn't that the same when people like get the flu or pneumonia when they already have those issues and they say, oh, well, you got the flu because your immune system was already.
Yeah, you can say that with flu too. But if you test for flu, they're going to put that on flu. They're going to say that person wouldn't have died today if they didn't have flu during flu season. This person wouldn't have died during COVID. They didn't have COVID during COVID season. So you look at it either way. So that lag phase is the time it takes the body to initially get the IgM up. Well, IgG is coming too. So it's not like we got to get IgM goes in there by itself. IgG is right behind it in the numbers of IgG antibody. So that's 10 days here, is what we're seeing. So this is our classic 10-day primary response. IgM comes up, IgG comes up, and then they level off. Okay? They level off and the illness is eliminated. Okay? Your antibodies made their move. They were developed, the B cells were stimulated, they went into plasma cells, the bone marrow maturation happened, the T cells were in the thymus, they went out. So all of that we talked about in the first unit is happening here, but these levels we can measure, okay? So we can measure these levels and we see that, and then it tapers off, okay? After 30 days, we go into the end, right? So we got lag phase, log phase, and decline. Then if it comes back, okay, the secondary response is the comeback, and it's going to be the lag phase. What you see here is it's not going to be 10 days. It'll be a much shorter time from the second exposure. This is where the vaccine principle comes in. So if we can shorten that lag phase here, say with a flu vaccine, We can give you a vaccine and get this response with flu vaccine. So we're going to put that antigen in you with a shot in the arm, and these two are going to come up. Okay, they're going to increase. But what you're wanting is this second exposure. When you really get the flu, when the flu patient sits in this room and sneezes, and you catch it, okay, and then you go home and you say, well, I didn't feel too good today. Well, that might be the only day you feel bad for flu, okay? But if you don't have the flu vaccine and you get exposed to that person that sneezed in the room, this lag phase is going on for 10 days, you're getting sick, okay? You're feeling bad, you're running fever, you're starting to drop off fatigue-wise, you're needing to get into bed. All of that is because you couldn't get this response quick enough. Your body had to suffer because there's no antibodies to help get rid of the flu. The virus is there, it's replicating, it's invading, it's going here to that cell, and you're sick, okay? So the whole idea of a vaccine is to shorten the lag, okay? The lag phase shortens down. We get a much stronger response from IgG because you've already got some memory, okay? And IgG pounces, and it's heavy in your blood, and as soon as that flu virus makes its entry, then your antibodies are starting to coat it and get on top of it, and then your immune response happens, okay? So that's long term. So this secondary response, this initial, leads us to memory of IgG, and we get that long term, ready to go immune. So another thing I wish they would talk about, I mean, I guess I'm just gonna have to do my own research, right? So what happened here? Why did I not get sick from COVID? Anybody want to guess? Anybody want to shoot it out there? Why aren't the majority of, well, we got 50 kids on campus now with it? Are any of them going to the hospital? I know some of them aren't feeling so good.
Exposed to it before?
Of some type.
Positive?
Some type of coronavirus, right? Coronavirus is not unique. We've got other coronaviruses that we've already been exposed to, okay? This one just happens to be a new one that infects animals, right? That never had infected humans before, that one. But what are the similarities? It's gotta be some.
Even with PPE, do you think you're still exposed to a lot of things working in the lab?
No, not in the lab. The ones we worry about right now is the phlebotomist, okay? So the phlebotomist is the one getting exposed to the patient. So if you do phlebotomy, part of your lab, like most mine, wherever I've ever worked, yeah, it's part of it. You gotta go out and stick patients. If there's a phlebotomist in the room that's going and sticking the patients, then that's who should really be wearing the N95 when they're going to face with the patient. Nurses should be wearing N95s. Doctors, I know y'all don't believe this, but how long are they with the patient, right? No, it's your nurse, right? It's the phlebotomist. It's the patient in front of person, right? Respiratory, my God, let's give the patient a breathing treatment in the room. Who wants to be in the room with a COVID patient breathing through the updraft machine, right? Nobody, So part of the deal is let's get a test, right? Let's get that patient tested while they're there, and that would make it. We could do that. We could say, okay, we've got before you enter the ER, let's swab your nose and put your mask back on. You'll wait 15 minutes, and then we're going to put you either in COVID room 1 or we're going to put you in regular room 1. And then COVID room 1, everybody's in there with N95s and taking stuff, that. Because we see it right now, N95s, right? Are they reusing them? Yes. Why? Why are we reusing N95s? Because we don't have
Are they
enough.
sharing or reusing their own?
Reusing their own. Keep yours, like hanging it on like your coat up. Hang your lab coat and your N95 mask on the hook, right? Let's get enough N95. So, but let's get a test, so we're not wasting N95 on who? The regular patient, right? Regular patient, we don't need an N95. Well, they say, well, heck, well, we're starting to see things that we can do. Test for the antigen. Test for the antigen 15 minutes later. If it is positive, that is a COVID patient. If it's negative, we don't know, right? So then we have to use our brain and say, well, are they running fever? Are they coughing and hacking? Or are they just coming in with a stomach ache?
So you could do the antibody test, but if they like are first getting sick from it, you don't think it'll be positive?
The antigen test. Okay, so they're still swabbing noses, right? And they're taking that swab and saying, is the COVID-19 antigen, the phospholipid membrane antigen thing we located, right? The testing mechanism. Is it on that swab? And they can tell you in 15 minutes now. They swab my nose and they want to look for the RNA of the virus, if it's located there. That one takes an hour. That can't be done in certain places. I know NEA Baptist does it. They've got but do they have enough reagents? So we've done all of this issues with, I got it, the test, but I don't have the stuff. So then we're going to the antigen test because it's quicker and I got the stuff, but then we run out of the stuff and then we got to go. So that's the way it's been bouncing back and forth with lab. Okay. But yes, I've recovered. But I went to the wedding, right? What I was finishing saying was, people right in my face going, hey, right? So I get exposed again, right? But I'm hoping that I've still got enough IgG. So every time you get exposed to it, what's happening? More. Okay? Every time. All right?
Did you wear your mask?
No.
Oh,
I wore it into the rehearsal dinner, and as soon as we got in the little room, off it comes. Just it's the way we've been doing things, right? Went to the wedding. The wedding was outside. I wear a mask outside, right? The reception was under a tent outside. So, I don't know if y'all saw Kansas State's football fans. How'd you like that social distancing of their students, right? Let's get 80 students in one line, but have a row between each of those 80 lines. They're wearing their mask, right? But they weren't. They're not what I thought would happen. What I thought was happening was you have a small pod of people, and y'all could sit. But that's one pod. Then you're going to have to 6 feet before I get another pod down. 6 feet before I put another pod down. I think that's what they got going on over here for Saturday. I don't know. But they're trying to. That's why they put 12,000 people in there Saturday night.
they did.
They're going to. But you might be on the top row, second deck over there, in your little pod. At least you'll be there, right? But the students, I'm gonna look, see what our student section looks like, but how y'all pack in there like row after row. Kansas State did that. They went row, skip a row, skip a row. But everybody was shoulder to shoulder, all the way across. It looked almost normal. We're almost there, right? I'm like, man, if they get through that, a couple more weeks, we'll be back to normal. All right. So IgM, complement fixation. Complement's coming. It comes after this chapter, right? Or it comes after cytokines first, sorry. Agglutination reactions that we're seeing in microbes already present, but we saw this with what? Coagulation, right? Staph coagulation test for Staph aureus. We saw that was agglutination. That's a clumping of together. That's touchy, I got you, and we make it clump, right? So we do a lot of our testing, especially this semester, which is about all we're going to do. And then next semester, you get blood bank, you do the same thing. Opsonization, salting the antigen, making it easier to digest and get rid of. And toxic neutralization. So this is definitely a great multiple choice question for IgM. Is all the following except, y'all love those, I know. Put 3 of those in, take 1 out, put something silly in there. So that moves us to IgA. IgA, it's about 10 to 15, so it's about the same as IgM. IgM was 5 to 10, so of all circulating in the serum, okay? But there's something IgA can do, right? IgA kind of gets out of the circulation, but you got to be a secretor to do it. And not every one of us is a secretor. You'll learn that in blood bank, so I don't want to get too deep for you. But some of us can secrete immunoglobulins in our fluids, mucus, semen, breast milk, whatever we're secreting, we can put IgA in there, okay? But if you're not a secretor, then you won't have those there. So how do we tell if you're a secretor or not? Basically, look for the IgA in your secretions. That's easy, right? We put an antigen to IgA and see if it agglutinates and see if you have it. But you got to be a secretor, not all of us are. So serum IgA appears as a monomer with a molecular weight of 160 Daltons, okay? But the IgA that we really pay attention to is the dimer of IgA, and that's the one that's in secretions. Okay, so we're going to do some subclasses here. Subclasses of IgA, IgA1, mainly found in the serum. So it's circulating with IgG and IgM. And this is key for IgA. It's an anti-inflammatory agent. Okay. We hadn't really talked about sensitivity, hypersensitivity reactions yet, but when we think about allergic reactions, we're going to talk about how IgA works there. It downregulates IgG-mediated phagocytosis, chemotaxis, bacterial cytoactivity, and cytokine release. So everything IgG is revved up to do, because IgG is our memory, IgG is our biggest responder. We have 80% of our immunoglobulins are IgG circulating, right? IgA1 downregulates that, slows down IgG. IgG could get crazy, right? So IgA has to downregulate it and bring it back to soft, low. Let's don't kill the body just because you've got an antigen you're chasing. The IgA2, which helps because it's 2, and this is the dimer primarily found in secretions. Along the respiratory, anything we breathe in into our lungs, we got IgA present, okay, in the mucus, urogenital tract, intestinal mucosa. I don't know if y'all think about what you eat, but you know, this is a great way. I love doing this. So when we eat something, right? Goes in our mouth, goes in our esophagus, goes down to our stomach, right? That is still external. I don't know, it's internal to the body, but it's still external to the body. It's a tube. All that's still external, so all that has to absorb through. Okay, so it'd be great during that mucosa that has some IgA present, because that's still external. Okay, the antigen hadn't invaded yet. It's in our stomach, might be causing some issues, right? But until it is absorbed, then it becomes something. So when you think about what you eat, like, you know, drink, you know, and we're in farm country, and you see these farmers out doing what? What do they do? Spray, 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 throw out chemicals, spray, 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 chemical, chemical, and all that gets absorbed in the water, right? So drinking water, eating food, we're still taking stuff in, so we still have antigens, okay? But this IgA being present in that intestinal mucosa keeps it from penetrating into the body. So until it absorbs, like getting something in your hands, right? While we wear gloves. Think of that. I think of back to the farmers I grew up with, right? They had a sink in our little drugstore, and they would come in after they poisoned their fields with some kind of chemical, and it would stain their arms. They wouldn't even wear gloves. They would have stained chemical arms that they would wash off in the sink, right? And that's absorbed in. So I'm trying to give you just this awful stories, but it's still external. So we have IgA placed in certain areas to keep us from taking in a respiratory antigen, right? Located where its entry is. All of these are entries.
What about just bacteria from food and stuff? Does that help with that?
Yes. Once it goes in, if the acid of your stomach doesn't kill it, it's going to get in the intestine and get absorbed in.
What about, like, I guess your saliva and stuff? Does it not?
It's look, all saliva has IgA. Yeah. Any secretions. IgA2 is more resistant to some bacterial proteinases, which makes sense, right? That are able to cleave IgA1. So regular stuff, a bacteria would say, "Ooh, I got something for you," right? IgA2 is a little more resistant to that enzyme that the bacteria produces. Little crossover there between Tuesday and Monday. All right, so here is the secretory IgA. Remember, if you're not a secretor, you don't have this. It's synthesized in plasma cells, found mainly in mucosal-associated lymphoid tissue. And so here's our plasma cell producing our IgA dimer. So it's IgA by itself, and then you get a J chain here, and it links it, okay? And then we have epithelial cell with a receptor on it. So the IgA does what? It gets linked into the epithelial cell. So this is that mucosal lining, endocytosis. So our IgA gets taken in. So there is a transcytosis. So once it gets in the cell, there's another movement through, right, until it gets out of that cell. And we have the IgA secretory component, okay, in the mucosal, past the mucosal lining, ready to sit there. It's released in dimeric forms, captured by secretory component of the epithelial cell. There's a binding. So this is great, right? So that's a little. And what does it function? It patrols mucosal lining and acts as the first line of defense. Great. We want that. We need that. We'll give you a minute. You need me on that wall, right? For the movie buffs in the room. Patrols, neutralizes toxin produced by the microorganism. It prevents bacterial adhesion or adherence to the mucosal surfaces. So it's great. Part of the reason we can take a respiratory pounding of somebody sneezing or coughing in our face, and we're like, I'm good, right? Because we're hoping that once we breathe that in, we've got IgA kind of hanging around and give us some defense to that. Passively transfers immunity to the newborn during breastfeeding. So this one is in breast milk. That is the transfer, not IgG out of the mother's breast milk, but IgA, so it's secretory. Okay. Complexes of IgA and antigen are easily trapped in that mucosa where they were found and eliminated by the ciliated epithelial cells. Now, here's my what do you want to call it? What are those little announcements that they put on, like for your good, like little health things, like wash your hands during flu season? Oh, a moment health. I don't know. There's some other thing I'm looking for. This can be knocked out. You might know how. You're vaping and you're smoking. Okay. Ciliated epithelial gets destroyed by cigarette smoke. So when you meet your cigarette smoker in your ER one day, and they're carrying their little oxygen tank with them, and they can't catch a breath without having to cough something out, and it sounds like they got to give it everything they can do to cough it out. That's part of the issue. The ciliated epithelial, the ciliation is gone. So there's no movement, okay, out of the respiratory tracts of that mucus where we caught that antigen, right? So that patient is more susceptible to pneumonia That's patients more susceptible to any respiratory illness because they don't have a way of moving that out. Okay, it was attacked, it was trapped, but it wasn't eliminated. Okay, so that's my don't smoke. Aggregation of IgA immune complexes may trigger the alternate complement pathway. We're coming to complement, so it's another one of those, hey, you got something coming. There's different ways complement gets activated. There's actually 3. And we're going to go over all 3. This would be one. The IgA would be one way of complement activation.
Are you talking about a TED Talk?
A little, no, not as serious as a TED Talk, but not as long either. You know, you have to do them, like each TV station has to have so many hours of these little, just so you know kind of things. I can't remember. There's a name for it. Secretory IgA functions. Neutrophils, monocyte macrophages possess specific receptors for serum and secretory IgA. Makes sense. Okay, because that's great, because any immunoglobulin that can attack the antigen, we want that other end of it to be able to tack on to the phagocytic cell, right? That's what these are. If you don't remember those, hopefully you remember that by now. So the binding triggers the respiratory burst, degranulation, cells involved. Both forms of IgA can thus act as opsons, which is that opsonization, right? Salt, put salt on the antigen, and are promoters of phagocytosis. So this seems like review, but it may not be. You may be going, he's still saying words, I have no idea what they mean. If you're still that case, if you're in that boat, which always have somebody in that boat, maybe 2 or 3 people in that boat, you just need to list. And I saw it today. I was looking for some of my labs I've misplaced. And I found the old first lab for immunology where it's just review of terms. So if you're in that boat, let me know. And I can post that, put it in Blackboard, and all of a sudden it just pop up as kind of like a review. But it's just like major terms of some, immunology terms that we started with first chapter. Now we go to IgD. We introduced IgD, right, is young. We haven't known about IgD very long, right? So where it is, right, we know it's extremely scarce in the blood, less than 0.001% of the total, so it's hardly even there in the blood. Has a molecular weight of 180. The delta heavy chain has a molecular weight of 62,000 Daltons, appears to have extended hinge region consisting of 58 amino acids. So you already know about this. So what's the main reason we have IgD? There it is. It's on this list.
B cell maturation.
Yes, B cell maturation. You've already had a test over that. It appears, right? But it appears on the surface with the IgM is there and the IgD is there, right? Okay. Short half-life found on the surface of immunocompetent but unstimulated B lymphocytes. Appears second after IgM. We know that already. They play a role in B cell activation, but it does play a role in regulating B cell maturation and differentiation that we need for the B cell to do what? Become specific for its antigen, right? Hopefully you hadn't lost that point. We do have secreted in the serum. It does not appear to serve as a protective function. It doesn't bind complement. It doesn't bind to the neutrophils or macrophages. And it does not cross the placenta, because that would be weird if it did, because we've said the only one that does that is IgG. So this doesn't sound like the others that we've talked about. It doesn't sound like G. It doesn't sound like M. It definitely doesn't sound like A. So those wouldn't be associated with anyone except D. And we have one more, E. Plays a little bigger role. Still not a whole lot in the serum of E. It has an epsilon H chain, where it gets its name, composed of around 550 amino acids again, and is distributed over one variable or 4 constant domains. This E is produced by plasma cells that are located primarily in the lungs and skin. So you're getting a hint of what this one does. So this is not secreted, this is produced. So you gotta have a plasma cell focused in the lungs and the skin for location. It doesn't participate in complement fixation or agglutination or opsonization. It's incapable of crossing the placenta, but what it does, what IgE does, it attaches to basophils, eosinophils, and tissue mast cells. Yay. Right? And have y'all learned about these two right here yet? Not yet? You're still in red cell? Okay. Have you seen a basophil yet on your dip? You seen an eosinophil on your dip? No? You know what an eosinophil should look like on your dip? Zoom, you can answer too. I see Lexi and Rachel and Brandy and Tristan. Well, with the right stain, right, basophils stay really purple granules, darker than the neutrophil. And the eosinophil is a red. It's got red granules here after the staining. Okay. But allergic reactions is E. E is associated with allergic reactions. So lung and skin, right? We think about where we have allergic reactions. 2 adjacent IgE molecules on a mast cell bind to the specific antigen, and then that starts the cascade of the allergic reaction, which is the cellular events and degranulation of mast cells and release of vasoactive amines such as histamine and heparin. Okay? When we have allergic reactions, you take what? Antihistamine, right? Benadryl. Antihistamine, right? We're trying to, this has already happened, right? We're having an allergic reaction. Anybody know what heparin does?
Anticoagulant?
Yes, anticoagulant. Why would we want heparin if we're having a degranulation of the mast cells and vasoactive amines? What would it do? Increase blood flow, right? All right, so here is that mast cell right here with the receptors for 2 IgE molecules of immunoglobulins. They got the antigen attached here, then they have this degranulation burst from the mast cell, and that's where your granules of histamine and heparin are. So we have allergic reactions, and this is where we talk about IgE, allergic reactions. So we talk about type 1. Type 1, immediate hypersensitivity results. This is hay fever, asthma, vomiting and diarrhea, hives, life-threatening anaphylactic shock. Immediate, like boom, right? You get exposed. There's no delay. You're allergic. Okay, you're allergic to whatever you just got exposed to, correct? So we have that with type 1. Eosinophils, which when I asked you, had you talked about them yet? Remember, eosinophils, what color do they turn again? That'd be the basophil.
Oh, wait.
Eosinophils are the red ones you're going to be seeing on your smears. They play the major role with large antigens such as parasitic worms that cannot be easily phagocytized. Okay, so we get a lot of E's, and you got to keep that straight, where your IgE, eosinophil, Okay, parasitic versus allergic reactions, a lot of different things running around your heads with these different cells. All right. I think we're not going to spend a whole nother day on chapter 5, but we've got more chapter 5 left to do. Now I'm going to be nice and end it on time, which is at 10 till, which it is. And we will see you downstairs for lab at 12:30.
Hey, I have a question, Mr. Rector.
Yes.
You can hear me. It's about micro. Is the study guide you posted on Blackboard, is that the same one that you handed out in class the other day?
I cannot quite hear you.
Is the study guide you posted on Blackboard the same one you handed out in class?
Did I hand out a study guide in class?
No, you handed
Oh, no. The thing I handed out in class is a flow chart.
Okay, I got you.
Chart.
Thank you.
And it's on, I don't know if I put it on there or not, but it's a flow chart.
Okay, I'll come get it.
It's not, sorry, it's not on there. It's just a picture of like where you go if you're gram positive, gram negative. But it is used for a study, but it's a flow chart.
Okay, thank you very much.
Then there's a table on Blackboard that has the reactions of each one of the ones we talked about for test.
Okay, yeah, I got that one.
Then we have the big quiz that not everybody's taken yet, which is due tonight at midnight because test is tomorrow. There is no class tomorrow for micro. We will have lab though.
Right, I got you. Okay, thank you.
All right, no problem. Okay, I'm going to end that for them.